normal cells divide and multiply in a controlled manner. In the last program, we saw how a substance called epidermal growth factor can promote the division of certain cells by acting on receptors on the cell membrane. If something goes wrong with this hormone receptor process, can it result in the uncontrolled growth which characterizes cancer? That's what interests researchers such as Mike Waterfield.
Epidermal growth factor is a factor that causes cells to proliferate, and being interested in the control of proliferation in cancer, it is a natural molecule for us to study. Now, the receptor itself can be expressed in enormous numbers on particular tumors, and if we look at this slide here, we can see that using a monoclonal antibody against the receptor, labeled with a green dye, we can reveal on these tumor cells in cultures enormous numbers of receptors.
Even these large numbers of receptors provide only a tiny amount of material for analysis. But the latest peptide sequence analyzers are so sensitive that only a small amount of material is needed to be able to work out the sequence of small sections of the receptor protein. Having these sequence data, it's possible to manufacture those same peptides in large quantities from their constituent amino acids. In one day, this machine can generate a peptide of 10 amino acids, large enough to prepare monoclonal antibodies which, incredibly, will recognize that peptide sequence within the intact receptor. We then have an analytical reagent for probing EGF receptors. At Liverpool University, Waterfield's monoclonal antibodies have been used to trace the fate of receptors in tissue stimulated by EGF. Most microscopy procedures result in killing living tissue, but for the antibody probe to work, the receptor protein must survive intact. Samples as thin as 1/100,000th of a millimetre are needed for electron microscopy, and the only way to cut such thin sections and keep the tissue intact is to use extremely low temperatures. The microscope grids which will hold the tiny tissue pieces require special treatment. In this vacuum chamber, they're given a small electric charge which will help the tissue samples stick to them. A sugar-coated wire loop is then used to pick up the frozen tissue and attach it to the treated grid. The key step is now to react the tiny slice sitting on the grid with the monoclonal antibodies. These antibodies have been tagged with colloidal gold, which gives the reagent a pink colour. About 6 months' work went into making that red drop. After staining, the tissue sample is examined under the electron microscope. Where the tissue has been frozen immediately after the EGF was added, the dark-stained antibodies reveal the receptor sites on the cell outer membrane. In tissue which has had EGF treatment 20 minutes before freezing and sectioning, receptors have been transported inside the cell and can be seen in vesicles within the cell matrix. The receptors are then broken down in structures called lysosomes within the cell, and they can only be followed for about half an hour. It'll be several more hours before the cells actually divide, so there's an enormous period of time about which we still know nothing. But we do know the protein sequence of the human EGF receptor. It's a huge molecule, over 1,200 amino acids long. It has 2 hydrophobic regions, one at the amino terminus, which is thought to put the protein in the membrane, and the other right in the middle of the sequence. So what does this mean?
This suggests that the receptor straddles the membrane, and here we see a very naive model where we have an external hydrophilic domain, a transmembrane hydrophobic domain, and an intracellular hydrophilic domain. Having this sequence, we could do something that we previously hadn't been able to do, which was using a computer to compare the sequence against all known sequences derived from other laboratories. When we did this, it was quite remarkable that we turned up a large region of identity with a gene that had been sequenced before. In fact, this sequence came from a virus, and it was a virus that causes leukemia in chickens, named avian erythroblastosis virus. Now, the gene that was involved was an oncogene, and this is a gene that is specifically able to cause that cancer, the leukemia, in some particular way, by a mechanism which we really didn't have any clues about. Finding the similarity between the sequence of this gene and the receptor now gave us a new clue, because it said that a molecule involved in transducing a growth signal was also capable of causing a cancer in the chickens. So if we use our potato model to look at the structure of the oncogene protein, we see that a large part of the external domain is in this case missing, so that it can no longer bind the EGF, the growth factor. Now, the consequences of this could be that the receptor is unregulated, and we have a system that is now out of control and sending a continuous proliferation signal to the cell, causing it to divide continuously.
So perhaps the faulty receptor mechanism can explain the chicken leukemia. And could something similar occur in some human cancers? This oncogene story gives us a tantalizing insight into how alterations in the growth factor system may lead to uncontrolled cell growth and ultimately to cancer. Another system which has given us interesting information about normal and abnormal cell growth is blood.
For biologists interested in developmental processes and pathology of cells and tissues, blood has many advantages. The most obvious is that one can take blood from veins. It's very accessible in large quantities. Blood is made in the bone marrow, and it's also accessible by sticking a needle into the hip bone. We can obtain reasonable quantities of bone marrow for investigation in tissue culture outside of the body. Compared with other tissues, we know really a great deal about the way in which blood develops in the bone marrow. This is an enormous advantage in trying to understand what goes wrong in leukemia and other diseases of the blood.
Normal blood has only a small proportion of white cells, shown here as the larger cells with nuclei which stain red.
Leukemia means white blood. And you'll see why if we just look at a blood sample taken from a leukemic patient. On the right here is blood from a patient with leukemia that's been sedimented in a centrifuge. And you'll notice that above the red cells at the bottom of the tube are a large mass or band of white cells. What has happened in this patient is that excessive numbers of white cells have been produced in the bone marrow, have been released into the blood to give this white coloration that can be easily seen. Compare that with the blood on the left, which is from a normal donor. There are white cells in there, but in such small normal numbers that they're hardly visible when looked at in a tube in this way. The different types of white cells and red cells that we've seen in the blood smear down the microscope are all produced in the bone marrow from a common progenitor that we call the multipotential stem cell. And I've shown here in this lineage or pedigree diagram how that process occurs. Here's a multipotential stem cell, which is a rare cell occurring at about 1 in 1,000 cells in the bone marrow population. And it gives rise through a series of genetic events that are rather poorly understood to each of the cell lineages that we can see eventually in the blood, the red cells and the white cells. The first cells that are produced are progenitor cells, which are now genetically committed to one or other of these different lineages. Although they're committed, morphologically, down the microscope that is, they're really rather indistinguishable. Following that commitment step, each of these cells now has to undergo a series of further genetic events which result in the maturation of that cell, usually within the bone marrow, to produce the final mature functional cell that will be released into the blood as a red blood cell or a phagocytic cell, or in the case of the lymphocyte, as an antibody-producing cell. In the case of the T lymphocytes, the cells have to be processed also through the thymus. And the lymphocytes are rather special. They finish up with each clone of lymphocyte producing a different type of antibody molecule as part of the defense mechanism.
Throughout life, our bodies constantly make blood. The bone marrow this donor is giving will be replaced within a matter of weeks. In such a dynamic process, there are many opportunities for biosynthetic mechanisms to go wrong or to be interfered with. Such events are fortunately rare, but the consequences can be extremely serious.
Leukemia can arise in any of the 6 blood cell lineages, although in practice, leukemias of red cell and their precursors are rather rare. In adults, leukemias usually arise in the myeloid, granulocytic lineages, whereas in children, it's much more common to have leukemia of the lymphoid lineages. Now, the main childhood leukemia is called lymphoblastic leukemia. The number of cases a year in the United Kingdom is only about 2 to 3 per 100,000, so it's relatively rare. But still today, it's a major killer of children, other than road accidents. Now, what we believe happens in leukemia, and I can illustrate with reference to childhood leukemia and the lineage diagram. We believe that a genetic event occurring in a single cell in the progenitor compartment initiates the disease process. So starting somewhere around here. That genetic event, which is in the nature of a mutation in genes controlling cell growth, appears to block the maturation process, so that, in this case, the B lymphocytes are unable to develop normally within the bone marrow. But that cell or clone which has acquired the genetic change will still proliferate. And as a consequence, the clone will accumulate in this immature developmental compartment and will increase in size and gradually begin to fill up the bone marrow space, such that it interferes with the function of the other cells that are required for defense and health. And it's the compromise of normal bone marrow function that in fact produces the diagnostic symptoms of lymphoblastic leukemia in children and indeed other types of leukemia.
The leukemia cells are larger than the normal white cells, but it's impossible to tell which type of leukemia is present just by looking at the morphology of the cells in a blood sample. Once again, antibodies are extremely valuable.
This diagram here, I show 2 cells, 1 and 2, which, for the sake of argument, let's say are morphologically identical. Antibodies can be made that are specific for A, anti-A that is, and that are specific for B, anti-B. We can then use these antibodies to identify and enumerate these 2 cell types. The way that that is frequently done is to piggyback on top of this binding antibody an anti-antibody, and similarly over here. And in order to distinguish the 2 reactions in the 2 antibodies, we tag the anti-antibodies with different fluorochromes or fluorescent dyes. In this example, the anti-A or anti-antibody is tagged with a green fluorescent dye, and the anti-B is tagged with a red dye.
Here are 2 populations of cells identified in this way. When illuminated by ultraviolet light, the antibody-labeled cells fluoresce, and differences are clearly visible under the microscope. High-speed counting machines can also recognize these fluorescent cells, and this method of cell sorting is now an established analytical tool.
Now that we can distinguish different types of blood cell and different types of leukemias, one of the key questions is what genetic mechanisms and what particular genes are involved in initiating the whole process of leukemogenesis in the bone marrow. Now, an important clue to the possible mechanisms comes from studying the way in which lymphocytes normally diversify and develop in the bone marrow, and particular genes that they use as part of this process. As shown in the model here, T and B lymphocytes both undergo a really rather remarkable process of genetic reorganization in antibody genes, or in the case of T cells, antibody-like genes used for making antigen receptors. And we can see how this happens with this simple model of the antibody or immunoglobulin gene. The red beads here represent the variable region segments of a gene, separated by an intron from the D or diversity region segments, separated by another intron from the constant region segments. What happens appears to be a process of looping and deletion, so that one particular V region in one particular B cell clone will associate with the D region, and the intervening loop is then excised to make what is now a functional and transcribed gene involving a particular V region with a particular D region to make this a unique antibody for that particular B lymphocyte. Further splicing then gives us the final messenger RNA and the complete antibody protein itself. Now, in a normal B lymphocyte population, there'll be a whole variety of different combinations of V, Ds, and Cs to make the diversity of antibodies that we can all make. Each individual B-cell clone that's developed in the bone marrow therefore has a unique rearrangement of its immunoglobulin gene, as shown here in the model. Now, because leukemia starts in a single B-cell precursor, every clonal member of the leukemic clone will have the same unique rearrangement of its immunoglobulin gene, which serves then as a molecular marker for that diseased clone. And we can use that gene rearrangement as a specific marker for the disease. If we take a molecular probe which will hybridize to some sequence of the gene, and we tag it, here shown in the purple, with a radioactive isotope, we can detect the particular configuration using the Southern blotting technique. In this track here, we see the pattern with a normal blood cell population. One band represents the unrearranged gene in the various white cells and red cells. The normal polyclonal B-cell populations have their diverse rearrangements spread throughout the track to give only a faint or diffuse background banding. But you see in these tracks, the next or subsequent 4 tracks, the unique rearrangements of the leukemic cell DNA, showing in most cases 2 bands representing the 2 parental immunoglobulin genes, rearranged in a different pattern in each of the 4 leukemias. These bands then represent distinctive markers for the leukemic clone. How do these rearrangements in B-cell antibody genes influence the development of leukemia? Well, one important mechanism we now have some understanding of actually manipulates the rearrangement process itself, which has made the unique antibody gene. What appears to happen is that a growth-controlling gene, shown here in black, is removed or excised from its normal chromosomal location and becomes inserted into the rearranging immunoglobulin gene. Now, because this gene is now constitutively activated in the B cell and will remain activated for the life of the B cell, the growth-controlling gene here in black also becomes activated and is frozen, if you like, in the on mode. As a consequence of this unique rearrangement process, this cell containing the rearranged gene will now be compelled to grow indefinitely and immortalized, rather than differentiating and maturing as it normally would.
The different types of leukemias can be given treatments appropriate to the particular disease. Many can be treated by chemotherapy alone, but in some patients, a bone marrow transplant is necessary. This woman is receiving total body irradiation to kill off entirely her own diseased blood and bone marrow before she receives a transplant from a matching donor. Irradiation is a grueling and traumatic experience.
Can you remember the period when you had the total body irradiation?
Oh, yes.
Now, what are your memories of that?
Just laying very still in a room, and it was a bit eerie, you know. Everyone goes out the room and you've got these cameras on you, watching that you don't move. And to keep still for 6 hours, 6.5 hours. But it's forgotten, that is easy, really.
Sheila Hills had a successful bone marrow transplant in 1985.
The strategy of treating the leukemias depends on the exact subtype that the patient is suffering from, and it varies. In the case of Sheila, she has acute myeloblastic leukemia. And what we do is we give drug treatment that gets rid of almost all of these cells, and the patient repopulates with their normal bone marrow cells. And they appear to be normal, except we know that there are a few of these cells still remaining. We have found it extremely difficult to get rid of those few remaining cells without having a profound effect on the normal marrow. And so the strategy we've devised there is to destroy all the bone marrow using total body irradiation. But then we've got to rescue the patient by giving bone marrow from somebody else, a bone marrow transplant. And this is very difficult. It requires a lot of support, but it is effective. Is your exercise tolerance all right? Climbing stairs and that sort of
It's
thing?
getting better.
It's getting better.
It's getting better and better.
And your hair's grown, hasn't it?
I know, everyone says how shiny it is. It's better than it was before.
Was it? I can't remember. Was it curly before?
It was wavy, but not so shiny.
Another strategy for some leukemias is to clean up the patient's own diseased bone marrow. First, the white cells are separated from the red cells. Monoclonal antibodies linked to a toxin called ricin and recognizing the leukemia cells can then be used to kill those leukemia cells, leaving the healthy white cells intact. The cleaned up cells will then be replaced in the patient's marrow.
How long basically does it take to recover fitness and down to a standard where one can, for instance, do a normal day's work first of all?
Because you're going to get your own marrow back and not somebody else's, this makes you get back to fitness much quicker because you don't have the complications of that other marrow attacking you, graft-versus-host disease. So I would expect you to be fit. You're an optician, aren't you?
Yes.
I would expect you to be fit for work in something like, say, 2 months after transplant.
Oh, as quick as that.
With Nigel, the subtype of leukemia he has is acute lymphoblastic leukemia. In children, this is very chemosensitive, and we can get the patient into remission. They repopulate with their normal marrow, and then we can give 2 years of treatment that gets rid of these few remaining cells. In the adults, this isn't so effective, and we've had to devise methods of getting rid of these few remaining cells that are more aggressive than we use in the children. One method is to give extremely intensive chemotherapy to the patient at a time when he is in remission. He'll need a lot of support for that. The other way is to have the patient in remission, take out his marrow, not all of it, just some of it, clean up that marrow to get rid of these few remaining leukemia cells using a monoclonal antibody directed against things on the surface, give total body irradiation to the patient to destroy all the rest of his marrow, and then transplant back into him his own marrow, an autograft.
The integration of Ray Pohl's clinical practice with Mel Greaves' scientific research has certainly improved leukemia diagnosis and treatment. Monoclonal antibodies which can recognize and, in the case of some leukemia cells, kill particular cell populations have been an invaluable result of basic research. But until we understand in more detail the mechanisms of cell growth in all types of tissue, our prospects for controlling abnormal cell growth elsewhere will remain limited.
We live surrounded by microorganisms which would rapidly cause our death were it not for the complex network of cellular activity known as the immune system. In this programme, we shall examine the discoveries relating to one part of it. Immunity is mediated by 2 interacting networks. Cells like this macrophage belong to the more primitive network, which mops up antigens such as bacteria or viruses, but can't distinguish between one antigen and another. We're concerned with a more highly developed network, the so-called adaptive immune system, which is mediated by white blood cells called lymphocytes. Each lymphocyte is able to recognize just one antigen out of the hundreds of millions that may enter the body. Experiments show that interactions occur between different types of lymphocyte, providing a complex system of checks and balances. Stem cells in the bone marrow give rise to two types of lymphocyte, T cells and B cells, which have different functions in the adaptive immune response. An antigen which enters the body triggers the adaptive immune system by stimulating T and B cells which are committed to respond to that antigen alone. When it activates a specific type of regulatory T cell called a helper, the helper in turn stimulates a B cell to start producing antibodies, also called immunoglobulin molecules. The B cell produces only one kind of antibody, specific to that particular antigen. The antibodies bind with the antigen, which facilitates their disposal by the body. When the antibody production is no longer required, another type of regulatory T cell, called a suppressor, switches off the B cell. So each B cell is part of a trio with a helper and a suppressor T cell, all responding to the same specific antigen. Yet another type of T cell, the cytotoxic T cell, has the ability to recognize specific antigen on the surface of both bacteria and infected body cells and destroy them directly. Embedded in all cell membranes are numerous proteins, carbohydrates and glycoproteins, some of which serve as labels or marker molecules. These marker molecules identify the type of cell this is, a macrophage or a liver cell or a B cell perhaps. Other molecules identify the species from which the cell comes. And there's also a set of molecules that are unique to the individual of the species. And these are called the Major Histocompatibility Complex, or MHC. And some of these MHC molecules appear on every cell in the body. Now, the MHC is as unique to an individual as a fingerprint. For example, it's certain that my MHC molecules are different from yours. And unless you have an identical twin, it's equally certain that your MHC molecules are different from those on the cells of every other living creature. Now, it turns out that the MHC molecules have a crucial role in the interaction of T cells with antigens, and we'll be coming back to that later. Now, all these surface molecules don't simply act as passive identification labels. They're also receptors, which can convey information into or out of the cell. So the obvious question is, how do they do that? How do they interact with other cells and molecules in the body? Well, more than 20 years ago, it was discovered that B cells interact with antigens by carrying embedded in their surface membrane a molecule of antibody with the combining sites for antigens facing outwards. Antibodies are probably the best understood molecules in the immune system. With a molecular mass of 150,000, they are even large enough to be seen by an electron microscope. They appear to have approximately the shape of a letter Y. The backbone skeleton of the molecule bears out this general impression. The overall structure appears complicated, particularly when all atoms are displayed in a space-filling model, but both chemical and structural studies have revealed an underlying pattern. They show that the molecule displays a simple regularity. It is composed of 4 polypeptide chains connected by disulfide bonds. The long chains are known as heavy chains and the short ones as light chains. Both are made up of repeating units or domains. In the case of immunoglobulin G, or IgG, the most abundant class of antibody, there are 2 in each light chain and 4 in each heavy chain. The domains show a great deal of similarity, both in amino acid sequence and in 3-dimensional structure. Those which do not vary from molecule to molecule are called constant domains and make up the main part of the antibody. The others, which make up the tips of the Y, vary slightly for each specificity and are called variable domains. It is these variable domains, one from the light chain and one from the heavy, which contain the binding site for the antigen. They are folded into two grooved barrels, which lie side by side and form the framework of the binding site itself. The sides of the barrels are made up of strands of regular sheet structure with interconnecting loops. The inner walls of the light and heavy chains, shown here in red, make up the sides of the combining site cavity. Some of the loops which protrude from these beta sheet walls vary greatly in sequence and sometimes in shape from one antibody specificity to another. It is these hypervariable loops, 3 from the heavy chain and 3 from the light, which determine the shape and chemical nature of the binding site, and therefore the type of antigen it recognizes. It's easy to see what an economical and yet adaptable structure this is, entirely built from repeating constant subunits, with the variability necessary for antigen recognition confined to a relatively small part of the molecule. Now, the genes underlying this structure turned out to be far more interesting than anyone could have predicted. The genes that code for the constant domains are common to all B cells. But the variable domains have to be produced in more than 100 million different conformations in order to combine with all the antigens that we know exist. Now, there simply isn't enough DNA in the cell to have a single gene for each one of these different conformations. The diversity is achieved by rearranging a relatively small pool of genes. Originally, the DNA contains multiple alternative genes coding for the variable domains of the antibody molecule. The DNA coding for the antibody light chain contains about 300 different variable or V genes, each capable of coding for the variable regions, plus a few joining or J genes which also contribute to the structural variation. Further downstream come the constant domain or C genes. Early in development, each B cell selects a V gene at random, and the DNA which lies between the chosen V gene and the J genes is excised by enzymes and the DNA strand rejoined. The DNA sequence from the chosen V gene to the C gene is transcribed into messenger RNA. Next, enzymes trim the RNA even further by excising all but one of the sequences transcribed from the J genes. This leaves a unique combination of V and J gene transcripts. Finally, the messenger RNA is translated into a protein light chain with a unique variable domain. A similar process of gene rearrangement occurs in the heavy chain. The idea that genes could be rearranged to generate diversity within antibody molecules was a revolutionary one. It was first proposed by Susumu Tonegawa, now at the Massachusetts Institute of Technology. He recalls the conceptual shifts that had to be made before his ideas could be accepted.
Any biologist thought that genes are very static in the lifespan of individual. So the idea that the genes rearranges during the B cell development was against that dogma. That's one. The second is there is this another dogma in molecular biology or genetics, which says one gene encodes for one polypeptide chain. Okay, so the idea of the variable region and the constant region encoded by separate genes sounds like there are two genes for one polypeptide chain. Okay. So that was the second point, which were not necessarily accepted by mainstream of biologists. So our finding went against both of those. That's a conceptual difficulties. But once it was shown, since the experiment was so direct, experiment data was so direct, it was quickly accepted by many.
When the genetics underlying antibody diversity was finally unravelled, it seemed reasonable to expect that the other family of lymphocytes, the T cells, would also recognise antigen by carrying an antibody-like molecule in their surface membrane. But despite years of exhaustive research, no such molecule was found. Whatever the T cell receptor for antigen is like, one problem in studying it is that T cells, unlike B cells, don't secrete large amounts of their receptor, which can be isolated and purified. Another problem is that T cells recognise antigen in a much more complex way than B cells. T cells can't recognise free antigen, but only antigen that's presented to them on the surface of another cell. Moreover, it has to be an association with the major histocompatibility complex, the MHC molecules. The question of how T cells see antigen plus MHC is still not resolved, but a major breakthrough in immunological techniques produced rapid progress. It became possible to induce individual T cells or B cells to divide continuously in tissue culture, each one giving a clone of identical daughter cells. Now, if a B cell is cloned, all its daughters secrete large amounts of identical antibodies, all binding with the same antigen. These are the so-called monoclonal antibodies, which have revolutionized many areas of biochemistry. Now, T-cell clones can also be made, and similarly, each member of the clone carries an identical surface receptor for antigen. Once identical T-cells were available in large quantities, it became possible to start work on trying to characterize the structure of the T-cell receptor. At the Dana-Farber Institute at Harvard Medical School, a group of biochemists and immunologists began to use these new techniques to study the T-cell receptor. Just as Susumu Tonegawa had to overcome the accepted dogmas in unravelling the antibody structure, so Ellis Reinherz, the leader of this group, faced a confusing array of theories about the nature of the T-cell receptor.
The current thinking at that time was rather confused. Some people were of